Welcome to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey of KISS Organics. This is the podcast where we discuss the cutting edge of growing from a science-based perspective and draw on top experts from around the industry to share their wisdom and knowledge. This week's podcast is with plant pathologist Chris Hayes. Chris began his journey into biologicals while working on his PhD, then continued in the field while a scientist at Cornell University. At Cornell, he worked on the strains of Trichoderma, T22, and G41 that would ultimately become the active ingredients in RootShield Plus. He left Cornell approximately 25 years ago to help start BioWorks. For the past quarter century, he has worked at various areas within BioWorks with the ultimate goal of helping those interested in using biologicals to understand the do's and don'ts to maximize the effectiveness of biologicals in programs to help prevent plant sickness and loss. Now on to the podcast. Hey, Chris, thanks for coming on the show today.
Thank you for having me, Tad.
Yeah, Chris, can we start off talking a little bit about your background and let people know kind of who you are and go from there?
I sure can. Hello, everyone out there. My name is Dr. Chris Hayes, and I hail from the farmlands of central Illinois and always had an interest growing up looking at alternatives to using synthetic chemistries to try to grow healthy plants. And I pursued that idea in both undergraduate and graduate school. And when I was graduating with my PhD from West Virginia University last century, I was looking into pursuing the idea of alternatives to synthetics to protect plants. And so I accepted a postdoc at the New York State Agricultural Experiment Station, which is governed by Cornell University. So I went to work at Cornell University in upstate New York in their field areas, working with different microbes, mostly fungi, that attack hosts to see if they had applicability to commercial agriculture. And so again, back last century in the late 80s to mid 90s, I worked at Cornell in the area of biological control, and we developed some pretty interesting microbes that a decision was made back in the mid 90s to leave Cornell and help start a company that we now call today BioWorks. So got about 25 plus years of experience actually out there promoting and placing biologicals into commercial horticulture, agriculture, and now the medicinal plant market.
Yeah, I'm very familiar with your company. We actually, a lot of the growers that we work with are using a lot of your products. I think you guys do a great job. And I want to circle back to some of the products that might be best for cannabis here in a little bit. But before we do, I kind of wanted to talk a little bit about Trichoderma and mycorrhizal fungus. And I know that one of the things that you and I talked about off air last week was just best practices and some of the differentiation between different strains of Trichoderma. So could you just kind of talk about what is Trichoderma and what are some of the best practices from an agricultural perspective?
Trichoderma is a genus of filamentous fungi found in the soil that have evolved for a purpose of suppressing or possibly protecting plants against pathogens. And I'll dwell more upon that in just a few minutes. But these are filamentous fungi that are found in just about all soil types from around the world. And their role is to do one of various functions within the soil. Some of them may just grow throughout the soil, taking up space. Others may grow throughout the soil, producing metabolites that may be both beneficial or harmful to both the plants and other microbes in the soil. So they're very diverse, these trichodermas. The name Trichoderma is very familiar in the growing of plants because there's been a lot of publications for a number of decades on trichodermas, and they've been isolated from suppressive soils that seem to have suppression to certain pathogens. Scientists have went out and dug up different strains of Trichoderma, different species, and have been able to identify some that, have the ability to help plants, not hurt them.
So what I'm hearing from you is that when we're talking about Trichoderma, it's a numerous species of fungi, and so we actually have to be very specific when we're looking at a particular product or talking about a particular Trichoderma. And I know one product you guys carry is called RootShield. Can you talk a little bit about how that product came about and what that particular strain is? Is it strain or species with fungus? I've always thought of it as a species, but I want to make sure I'm using the right nomenclature.
Trichoderma strains, Trichoderma strains are like different family members. Uh, some are really strong and good, and some can be a little bit naughty. So it depends upon the strain of Trichoderma, how strong or how weak, or what they can do of any value or have no value at all. Because the group, the genus Trichoderma, they're known for their abilities to be found in soils around the world, and they're workhorses out in the soil. But again, sometimes they do good things and sometimes not so good. So they are very strain dependent. And we took that concept to heart at Cornell University. When we obtained a grant, we were looking at, can you produce a hybrid strain of a Trichoderma that may be able to be used as a biocontrol agent in a commercial agricultural use. So with this grant at Cornell in the lab of Dr. Terry Harmon, hybridization studies were conducted. Now these are asexual fungi, so protoplast fusion was used to mimic what happens out in nature to produce genetic variability. And so strains were selected from various areas in the U.S. where disease suppressive soils have been identified. And what I mean by that is farmers would report record yields, high yields in their soils, but their neighbor across the street using the same inputs, the same genetics, inputs having the same basic soil profile did not have the same success. And scientists will go out and collect samples from these different soil types and try to identify something that may be unique in these suppressive soils. And that was done. And a couple of different strains were identified from suppressive soils. Hybrids were made using a process called protoplast fusion. And then time was spent to look at these different hybrid strains to see if a superior hybrid could be identified that may have commercial attributes.
So that's where RootShield came from, the T22 strain that you manufactured?
The actual number T22 of the strain, its strain number, is called 1219-22. Strain 12 was inbred line was fused to strain 95, and many different hybrids were isolated, dozens and dozens and dozens, over 100, and it was the 22nd hybrid cell isolated under the microscope when we took it for further testing had the greatest attributes to attack the greatest range of pathogens. It would grow on the entire root system. It would grow in soil pHs, soil temperature, soil moistures that were highly variable. So this strain had attributes that made it pretty strong out there in the environment against a range of issues in a variety of environments. And because the parents were chosen from suppressive soils where synthetics were used, this strain had a high degree of compatibility with many chemical fungicides that developed resistance. And so this was the original strain or active ingredient in our product, RootShield. But being scientists, you always look for what else is out there that can help. Being businessmen, you always want to have something interesting to talk about, and you always want to try to improve your products to offer more efficacy to the farmers and the growers out there in the real world. And so we had some single line isolate strains that were pretty strong and had attributes in excess of what the hybrid strain T22 had. One of these strains was classified as G41. It was a Trichoderma virens, and it had the ability to grow in very wet soils, and it liked to attack pathogens found in the group of the oomycetes, including Pythium and Phytophthora and downy mildew. So These this other strain was mixed with the T22 hybrid strain, the G41 strain, and we introduced a product a couple of years ago called RootShield Plus. The name RootShield was pretty good. The name RootShield Plus, we thought we'd keep it. So we now have two different strains in the same package, and they complement each other. And in fact, we were granted a patent on the synergy between these two strains, which means they act better together than either one would alone. They're a heck of a couple.
Yeah, so you mentioned some effects against NYSAES, which I'm not great at pronouncing either. What are some of the attributes of these particular strains in terms of the RootShield Plus that people in cannabis may see benefit from? You mentioned downy mildew, for example, and I want to talk about the mildews later in the podcast.
Yes.
But what are just some general attributes associated that you found from these particular strains?
Well, these strains are what we call rhizosphere rhizoplank competent. They like to grow not just in the soil, not just near the roots, but they grab onto the roots and actually coil around them, most importantly, root hairs. So when we use the name root shield, we're truly talking about shielding the roots, blocking entrance in the root tissue by pathogens. So that's one of their strong attributes. They cling on very tightly. And so, you know, if you're growing clones, either from cuttings or seeds, you know, you want to protect that newly emerging root tissue. And these trichodermas found in RootShield Plus will do that. We have a wettable powder you can drench with. We have a granular you can incorporate or even top dress and water down because they grow. So these are little microbes, fungi that will colonize and protect that root. And that's a pretty good thing, shielding it. In addition, these fungi produce enzymes. The T22 strain produces an abundance of what we call chitinases, and many of your fungal pathogens that try to attack the roots contain chitin as a sugar moiety in the cell wall. Other pathogens do not contain as much chitin in their cell wall. They contain a sugar moiety called a glucan. And I don't want to bore your audience with the chemical structures, but they're close, but a little bit different. A little different spin on a glucose molecule. And this glucan, we have that additional strain, the G41, that produces an abundance of glucanases. So it attacks different pathogens by dissolving the cell wall and feeding on it than the T22 strain. So together, they produce these enzymes, these chitinases and glucanases that will attack the sugar moieties in most fungal cell walls. So these trichodermas not just cling to the roots, but they'll actually attack pathogens that get near the roots and stop them from entering root tissue. And in fact, we have seen studies on fungi that produce survival structures called sclerotia. Sclerotinia is one, and our friend powdery mildew produces sclerotia, and Botrytis produces sclerotia. My point is, these fungal pathogens produce survival structures in the soil that allow them to hide from synthetic chemistries. But the trichodermas can sense where they are. They'll grow to them, wrap around them, and start to digest and feed upon them. So these trichodermas found in RootShield Plus can lower pathogen levels in the soil. And that can be advantageous because if you attack pathogens, especially those that may grow out of the soil and attack the foliage, if you suppress them in the ground, you'll have less potential when they surface out of the ground to possibly attack the foliage and flower buds.
Yeah, now, in a healthy ecosystem, are you seeing persistence with these trichoderma strains? Are they lasting, or is this something that needs to be applied on a fairly regular schedule if you're doing intensive cropping like you would be with cannabis?
Well, I know out there people listening would love to have something that lasts forever, but the EPA says that's a no-no. We don't want you to release something that's invasive, that will take over. So what we see with our trichodermas is that they'll colonize the root tissue in a protective level for 8 up to 12 weeks, depending upon the organic level of the growing media. Organic levels tend to encourage these trichodermas to persist. But after 12 weeks, we see these levels begin to drop, and by 16 weeks, there's no appreciable protection with these strains of trichoderma found in RootShield Plus. So it doesn't last forever. And the other interesting point is that they have compatibility, we know, with mycorrhizae. Golly, back in the 1980s, late 1980s and early 1990s, we were doing work with the USDA, Dr. Bob Lumsden, who was able to show that our strains of trichoderma had no negative effect, or little or no negative effect on mycorrhiza. And Dr. Mary Hausbeck from Michigan State University also published and saw less than a 10% or about a 10% reduction in mycorrhizae concentration when using these strains of trichoderma, which is not significant. So we don't always know why they're compatible, but when we place them together, they seem to not have a negative effect on each other. So that's a good thing, but I like to say that doesn't hold for all the different strains and all the different species of trichoderma that may be offered to you out there. Like I said, a lot of these strains may produce metabolites that have a negative effect on plants, or may have a negative effect on mycorrhizae. That's why some people say you can mix trichoderma and mycorrhizae. Others say, I don't know. Others say you can't. It depends on the strain. And that's very important to understand. Not all strains of trichoderma are the same. Not all humans are the same. So it's important to understand the strain and ask the questions. Hey, company, do you know what your strains are compatible with?
I'm glad you highlighted that, because this is something that you and I talked about last week as well off air. And for me, one of the challenges I see is I see these commercial products, and here's one that I'm just looking at the label. It has 83 propagules per gram of Glomus enteratices, Glomus masiae, and then 313,125 propagules per gram of trichoderma. So you have a preponderance of trichoderma in there at concentrations, exponentially higher than the amount of mycorrhiza in the product. Even though the product is marketed as a mycorrhizal product, and the company claims that these things play nice together, but we don't really know, as an end user without seeing a lot of research to support it. So I just want to caution listeners to do their homework and realize too, what's on the label isn't always what's actually in the product too. That's a whole nother challenge that we're finding. The Oregon Department of Agriculture has been testing a lot of products and finding that with microbials, you don't always get what you think you're getting when we're talking across the board here. But I have tried RootShield before. I think you guys do a great job with your products. How does that work in terms of shelf life with the RootShield and RootShield Plus? Are people able to walk into a store and purchase it? I know I ordered online and had it shipped directly from you guys to me, and this was quite a few years back. But how does shelf life and those sorts of things, stability, come into play with a microbial product like this?
Well, just to make sure everybody can find this information on their own, please check out our website, www.bioworksinc.com. Because on there we have compatibility information, shelf life and storage information. If you want to treat 10 plants or 10,000 acres, we have small volume and large volume calculations for you. We have a very high quality expertise staff. So ask the expert area on our website, send in your questions, give us a call. We have a lot of PhDs. We have 100 plus years of expertise and experience with some of the people we have in our technical services and our biological program managers, along with our people like myself, our biological solutions advisors. We throw a team effort at trying to give you an answer. But to answer your question quickly, with our products, it's temperature dependent. Most trichodermas, they are temperature dependent. So if you store it at room temperature, you can get 6 months. In the refrigerator, you'll get longer. In the freezer, it stores even longer. So we have a a shelf life table based upon temperature. Can you walk into a store and buy our products? There is a chance you can. However, we typically try to direct people through our distribution channel. And again, on our homepage, if you click at the top, or if you go to the top, there's a button where to buy, enter the state, enter the country, because we're in multiple countries, and you will see a listing of our distributors and a way to contact them. So we try to do it that way. We do have a retail site. We have a couple of retail outlets, Johnny Select Seeds, Arbico, and others that you can order from. So we try to make it easy to get it from you, or get it from us, to get it to you. We try to give you shelf life information, tables that you can print off. They're all PDFs. We've got compatibility with other biologicals, fertilizers, adjuvants, and other synthetic inputs. I mean, here are fungi that you would expect not to be able to be mixed with chemical fungicides, but they can be. There's many you can. Because the premise behind biologicals, RootShield Plus, and anything else is you want to prevent, you don't want to try to cure. And in cannabis and hemp, your toolboxes are limited in what you have available. And so you want to prevent and not have to go to that toolbox for a hammer, because you don't have a lot of hammers. But prevent, and not only preventative from a root protective aspect, but preventative from a foliar aspect. You know, things can get out of hand real fast above the ground. And if you don't have some preventative out there, you're going to lose some plants. You're going to lose some flowers.
That's a good segue into the next part of our conversation. But before we do, I had one last question about Trichoderma, specifically the ones that you are offering. Do those offer any assistance in regards to the microbial loop or nutrient cycling in terms of plant health or plant growth beyond the disease resistance potential?
How much time you got, Ted? Oh, we published a paper out of the station called Solubilization and Sequestration of Inorganic Nutrients by Trichoderma strain T22. Don't ask me to say it again. And to answer your question, not just yes, but heck yes. Our trichodermas have the ability to solubilize insoluble nutrients. We would take culture filtrate from strain T22. We'd put rock phosphate in it, put it on a shaker overnight, come back in the next day, and it was gone. Somebody stole the rock? No, they didn't. It was dissolved, because our trichodermas produce reducing agent, chelating agents. They're part of the recycling of food back to the root tissue. In addition, and this is a published paper, in addition, another paper was published out of the lab from a scientist visiting from Taiwan, again, last century, and they were able to show that utilizing our trichoderma strains as a soil drench, that they could inhibit viral uptake by the plant tissue. I got a paper in front of me right now I'm taking a look at called Trichoderma hartzianum. T22 induces systemic resistance in tomato infected by cucumber mosaic virus. We're seeing these trichodermas can turn on what's called induced systemic resistance within that plant. It helps produce things like phytoalexins, which are the antibodies of a plant. It's quite fascinating. In fact, we did a study, again, a while ago at Texas A&M on cotton seedlings out of the strings, and with T22, we saw a 10-fold increase in phytoalexin production in the roots. And again, the phytoalexins are one of your plant hormones that are produced as a response to a problem, an infection. They're the so-called antibodies in a plant, and they move from the roots up. And so, to answer your question, yes, there's a lot of publications on this. If you would just search solubilization or mycoparasitism or induced systemic resistance in T22, you're going to see a lot of papers popping up. So there's a lot of publications. We try to make sure that we have data to support what we say.
That's great. I love that. I'm going to put some links up on the podcast page to your website, as well as some of the publicly available papers. You and I can talk about that after the podcast, and we can put some of those up. And then I'm excited to put you in touch with Dr. Ben Higgins too. and start maybe doing some research on cannabis here with some of these things, because I think that's really exciting what you guys are doing.
Ted, I've been talking to scientists around the country. We want to set up studies to see how metabolite production is enhanced, reduced, altered using our inputs. We look forward to having that data. We look forward to those studies. So please, anybody out there, we want to work and have data. We've strived for many, many decades to offer information, so you're not guessing. You're using facts and figures to guide your programs.
Yeah, I totally agree with you on that. I think that's wonderful what you guys are doing. You had mentioned some of the above ground challenges, which was something that I was really looking forward to talking to you about with your background in pathology. So one of the big challenges in cannabis is powdery mildew. Can you talk a bit about what it is, how it relates to cannabis, and how we might be able to better treat and prevent it?
Well, powdery mildew it's tough. It attacks cannabis. Powdery mildew is just a general name for many, many different genera that are included in the grouping powdery mildew, 30 plus, and they're finding more all the time. So it's pretty broad spectrum. It typically occurs on the upper leaf surface as a white powdery kind of hyphae, little hairs, and so it's unique in that it's an obligate parasite. So it likes living tissue to grow and develop. It's unusual in that it doesn't require free moisture to germinate. Things like Botrytis, with free moisture, the thing can go crazy quickly. This thing doesn't need free moisture. It can actually pull moisture out of atmospheric water, we actually call humidity. So it can get out of hand. It likes to thrive between temperatures of 60 to 72. If you can raise the temperature in your greenhouse or your atmospheric control unit to about 75, it just doesn't thrive real well. It's interesting in that powdery mildew needs an open wound or a cut or a stomate to enter leaf tissue. It differs in, Botrytis and Downy in that they'll make enzymes and dissolve their way through the cuticle and the epidermis and the leaf tissue. Powdery mildew really doesn't do that. So, you can use certain products, certain contact killers, certain hammers, and some people will even say that's a curative effect. Well, powdery doesn't enter quickly, so I don't know if it's curative. You just happen to whack it before it got into plant tissue. So it's an interesting fungal problem. To control it, just get the humidity down. Get it down to 45% if you can. I know that's pretty low, but that really discourages powdery mildew. That, or get the temperature in your greenhouse around 75 to discourage it. With any pathogen out there, Tad, that may harm a plant, it's always good to find the best genetics available. And I know there's a bunch of work being done in cannabis on genetics, because resistant varieties, there's nothing a total resistant, but boy, if you can lower pressure significantly, then some of your contacts and biologicals may be all you'll ever need to keep things from getting seriously out of hand if you use them preventatively. So it is a very common problem, powdery mildew. The multiple genera can probably attack over 1,500 different types of plants. So it's a very common problem.
So I have a few questions here. Let's start with do I need to worry about the powdery mildew on my comfrey or the powdery mildew on my squash infecting my cannabis plant in my garden if I was, say, in a greenhouse or outdoors? Or is there a specific species or strain that's affecting cannabis that's different than what we're seeing on these other vegetables or other crops? Or do you know?
Well, sometimes they can jump from one crop to the other. It just depends upon the strain of the pathogen. Some of these pathogens are more crop specific. Some of them aren't. So it's always best if you got other plants in there with powdery mildew, it may not be a bad idea to treat those plants. But again, it just depends.
So what I see, as someone who grows a fair number of vegetables with my squash and stuff, when I start getting the colder nights, I'll have like a really hot sunny day, and then the next day it'll rain, or I'll have a really cold night, some sort of water event, and then I'll start seeing powdery mildew a day or two later popping up, and the spore pressure just seems to get really high. And I can spray that pumpkin plant or squash plant with an oil-based solution that will, kill that powdery mildew, but it seems to just keep coming right back. 4 or 5 days later, I've got powdery mildew again. And with cannabis, I know you can't spray a lot of stuff in flower. I want to talk a little more about the humidity and temperature thing, but what are your thoughts in terms of treatment? So when we're contact killing powdery mildew, is it because of the spore pressure in the air that we're seeing more pop up? Like, do we need to control that, or is the plant just damaged at that point? It has easy access and it's just going to keep coming back once we've seen it on a plant. Does that make sense?
Pathogens are like the IRS. They never go away. They're always out there. They're always wanting to do something. It just takes the right environmental trigger to turn them on. And powdery mildews can survive up to 3 years in plant debris. So, they have persistence. Others can survive for decades, but other pathogens. But powdery mildew can survive in overwinter in plant debris and survive a couple of winters, up to 3 years in plant debris. So it's always out there. It's good to use, as you said, some of these contact hammers, especially when the environment, as you described, starts to favor the occurrence, because under certain environmental conditions and triggers, a couple of days later, it's happening. So when that trigger hits, let's get out the preventatives. You mentioned oils. Oils are a great contact. We have an oil. I will mention it. It's called Suff Oil X. The only reason I mention it, that if you've ever used an oil that looks like milk, that's Suff Oil X. It's an emulsified oil to give it charge so it will react with water, and it has some attributes from that aspect. But other things you can use besides oils for these foliar pathogens are soaps. Soaps can be effective, but be careful. They can weaken the cuticle and do other things. And then we have a salt-based chemistry called Milstop. It's a potassium bicarbonate that will desiccate and not allow fungal spores to either germinate or grow very well because of the desiccation properties and also the pH alteration properties of this product.
I've also done some experience with, there's an enzyme in milk, raw milk apparently too, that has some efficacy. I know that there's some specific biological fungicides as well, products like actinovate, but I haven't had amazing success with the like streptomyces and some of those other ones personally. I don't want to say that they don't work, but for me, a lot of these other applications, well, the milk, it dries and it looks like powdery mildew on your leaf surface, which to me was that was a deal breaker. But the streptomyces, I felt like I had to apply it like every few days. Like I wasn't getting very strong suppression personally. And this is purely anecdotal in my experience. The oils all worked really well. I haven't done anything with potassium bicarbonate. But at the end of the day, I find nothing completely wipes it out. Once I see it on a plant, I feel like I'm battling it until I crop. Have you had any success or any other experience with people having more success with that? And then if we're cloning that plant, what are the risks associated with, besides, let's just say, being a weak genetic line or poorly resistant to powdery mildew? Are there any other risks of re-inoculating it or bringing it back into our controlled environment?
Yes, there's always risks when you take. That's why on your mother plants, you should do your best. People sometimes think, "Oh, mom's fine. I don't need to worry about her." Well, mom needs to be taken care of too. And so, before taking cuttings, I'd encourage people to put some one of these products down. Salt, soaps, oils. And you mentioned streptomyces. Tad, have you ever made use of products that contain a bacillus? Different species, but a Bacillus subtilis. These bacteria, Tad, have you ever used a bacillus before?
I have. There was one I got from Wilbur Ellis back in the day, and I'm trying to remember the name of it, the commercial name. Maybe it'll come to me while we're chatting.
Oh, that was Bacillus amyloliquefaciens, or what the heck? Anyway, I know that product. I'm trying to remember too. Sorry, I'm having a senior moment. You're just, but and what I like to talk about is, and I'll make mention now is, I like the hammer residual concept. I like to mix these salt, soaps, and oils with a biological that may have some residual activity based upon its mechanism of action. And so, you are right. Some of these, hammers, the oils and the salts, they got to be reapplied because they don't last a long time. That's a good thing or a bad thing, but they don't have a lot of residual. So why not mix it with something with a residual? And there's some decent bacillus-based products out there. We happen to have one called CEASE. It's a Bacillus subtilis, strain QST 713, for those of you that want to know that. And got compatibility, got shelf life info. And these bacteria in general, Tad, their mechanism of action is to produce metabolites. That we call them sometimes cyclic lipopeptides and biosurfactants, siderophores. There's all sorts of names, but these metabolites have the ability to make holes in cell walls and cell membranes, and cells can bleed to death. They can collapse, and those metabolites can have some residual activity on the foliage. So you mix. In a lot of our programs, you'll see us recommend mixing our bicarbonate Millstop, which will help spread and stick the metabolites with CEASE, which has the metabolites, so you get a quick, direct kill on contact, and then you get a residual activity from the metabolites found in CEASE. So, that's where you can start putting these biologicals together in your programs for a quick kill and a residual activity, so you don't have to come out so often.
So I looked up the product. It was called Sonata. It's a Bacillus pumilis. I'm terrible. I'm terrible with these names.
Bacillus pumilis. Okay.
But one thing I wanted to jump back to was regarding powdery mildew and temperature and humidity. So one of the challenges, traditionally people would grow cannabis during the flower cycle and just immediately drop the humidity down into the 40s for that very reason that you mentioned, because the molds and mildews just don't enjoy that level of humidity. But I find what we found, and again, this is still fairly anecdotal, but by using a vapor pressure differential and raising the humidity relative to the temperature, the plant seems to be more resistant to these particular mildews than prior problems. So we've had some cannabis growers that have, rather than their traditional dropping the humidity, they've actually raised the humidity level in the rooms and raised the temperature. And by doing so, they've been able to reduce the amount of powdery mildew that they've seen expressed on the leaf tissue in cannabis.
I think that's a great idea, and I've seen people do that too here in Florida when I go visit, and elsewhere. And because as you raise the temperature, the humidity changes. But the most important thing is about 75 or higher, powdery just doesn't like to grow well. It just doesn't. So raise it up a little bit, even if the humidity goes up, because the temperature is still not conducive for this problem to get out of hand. And you remember the pathogen and insect and pest triangle. You need the host, you need the problem, and you need the right environment. And so raising that temperature, even if the humidity goes up, it's still okay because you're not going to allow, because of warmer temperatures, the powdery to get out of hand. So I've seen that work too. I agree with you.
So we've been starting with the higher humidity and then slowly tapering it down towards the end of flower to help prevent botrytis, because it seems like as the plant is starting to senesce, as we're getting later in the crop cycle, we're more at risk for botrytis as the flowers are developing. And it seems that if we don't drop that humidity towards the end of the cycle, we're going to still have some botrytis challenges. What can you talk about botrytis and sort of how it relates to powdery mildew and some of these other things? Because one of the issues here is we can't spray a lot of stuff on cannabis flowers in flowering cycles, and even the microbials are really challenging because some states are testing for microbials, but they're not differentiating between different types, gram-negative bacteria or all these different things. Just microbial biomass is all they're looking at. And so there's a lot of challenges around testing that just from a legal perspective make it really hard to treat these things if we don't get ahead of them early on.
Oh, no question about it, Ted. No question about it. And they'll do a total microbial fungi and mold and things. And they that's why, with some of our microbial living products, we recommend stop applying, before, harvest of flowers, because our goal is to ultimately go to these testing labs and set up primers and assays to allow distinction of what's there, so you can negate the use of some of these beneficial biologicals, because they are darn good tools that offer, nothing negative to the plants. Just all of my products have exempt from residue tolerance levels. So you don't have to worry about anything they produce as being toxic to you, the environment, or the plant. All of my products have at least one formulation organically certified. I mean, if you were growing something you could pick and eat, tomatoes and an apple, we have 0 pre-harvest intervals with our products because they don't produce anything toxic. But there's still testing going on to look at total microbes. So that's where you may want to make use of some of your non-living biologicals, some of these oils and salts and soaps, because what happens is when the flower closes up and gets nice and dense, it holds moisture in there, and that's where problems can get out of hand inside that flower. And I know you can't use bio, so before it closes too tight, maybe use one of these other inputs. That could be helpful. But the thing with botrytis is it differs from powdery mildew. Botrytis needs free moisture to germinate, so it likes it when it's wet out. So botrytis, you don't want to water plants at the end of the day and leave them wet all night. You'll have botrytis popping up the next day. You don't want to do that. So you want to keep your plants dry. So I know people that have issues with botrytis will space plants a little more so they can dry. Others will turn on fans to help dry plants. Now, you don't want to do that with botrytis because botrytis is dispersed by the wind. So when botrytis sporulates on the soil surface and you turn fans on, all you're doing is spreading it. So you got to be careful with airflow and botrytis. Whereas airflow can help with powdery mildew and other issues, it's not good to have good airflow because it can cause spore dispersal with botrytis. So they're just a little bit different. Botrytis will produce chitinases and lipases, enzymes that allow it to penetrate right through the leaf surface and not have to worry about finding an opening available to it. So it's a tough one. Botrytis is different from powdery in that, there's multiple species. Botrytis is the genus name, but there's multiple species. Fortunately enough, these species are more host specific. So as I mentioned with your powdery mildew, you got some cucumbers or something near some medicinal plants. I wouldn't do that. There's a chance. Not always, but there's a chance. Less of a chance with botrytis because they're more specific. But you've got one certain species, Botrytis cinerea, And I got 11 years of college on Tad, so I can pronounce these names pretty well. I better be able to. And this Botrytis cinerea, it's a beggar out there. It can attack over 200 different plants, both monocots and dicots. So it's a gnarly one. And you buy strawberries and you put them in your refrigerator, right? Why? Because I want to eat them in 2-3 weeks. Well, you can't do that. There's something gray growing on that. That's Botrytis. We call it gray mold. And, even at refrigerated temperatures of 46 degrees Fahrenheit, it can grow after time. So it likes it cool. It likes it moist. So it differs than powdery. You won't see powdery growing in your fridge. I can't imagine. So it's a little bit different. And on the good side, Botrytis doesn't survive for 3 years like powdery does. Botrytis, a couple of weeks to a couple of months, maybe a little longer, but and this is a neat one. And Botrytis is related to Sclerotinia. It's in the same family, and they form these survival structures in the soil. And at Cornell, we used to do studies looking at how well these sclerotias could be colonized by our different strains in RootShield Plus, and they did a pretty good job of growing on them and knocking them out. So that's where I talked about earlier, the suppressive nature of these strains to suppress pathogens in the soil before they can surface and do foliar damage.
So can you explain to me sort of the physical mechanisms associated with Botrytis? And also you touched on its life cycle a little bit here. Can you sort of explain how it survives and exactly what it does to attacking the plant?
Well, the survival structures are called sclerotia. They're these little, they produce hyphae and other materials that encapsulate the survival spores and the surviving cells inside. And when the right environment is there, again, it can be, they thrive from about 60 to 75 degrees Fahrenheit. So they thrive at about the same temperature range as powdery, but they can survive in much cooler environments. And again, you get enough moisture, a good rain, and you got these temperatures in the soil, they're going to come out of hiding. They're going to grow to the soil surface. The Botrytis is going to sporulate. The spores are going to be windblown. And that's how they move around in the environment. Botrytis also, as I mentioned, will go directly through the cuticle, through the epidermis into the plant tissue. But especially Botrytis cinerea, they actually produce small RNA molecules that can affect the plant's immune system. So some of these pathogens, when they attack, they'll turn the plant's defense system off. And they have free reign at that time. So did I answer the question, Tad? Like you?
Yeah. So I've seen Botrytis spread really quickly, take over through. And I've seen people attempt to treat it in a variety of ways, from everything from hydrogen peroxide to cutting off and wounding that particular bud and taking it away from the room. But at the end of the day, they all feel like losing battles. Once you've spotted it, you're kind of in trouble, it seems like. What would be the best way to address it, and how quickly can it really spread? It seems like it spreads like wildfire in my experience once it's established in a room. But can you touch on those things a little bit?
Yeah, I mean, if you got free moisture in the right temperature, the right environment, in 8 hours it can germinate. In 8 to 12 hours, so in half a day, if you got the right conditions, it's germinating, it's growing, and it's producing enzymes going right inside the leaf tissue. So you can't get it with contacts. So that's why when you either have a history or you start to see a few plants having issues, that's when you got to quarantine that problem and hit it with some contacts. Also, there are some biologicals that are available. Again, mix some of these hammers, these salt soaps and oils, with something living. Again, our NYSAES product is helpful, but we brought in a new fungus from New Zealand, Australia region, Tad, a couple of years ago, and started testing it. And it's a unique biological in that most biologicals either attack a host or produce metabolites that are detrimental to the target or other things. This fungus, and it's technically called Eulocladium udomonsi, strain U3, is the name U2 was taken. And this particular strain of fungus is called technically an antagonist. So when cells senesce or die, they rupture and they release moisture and nutrients. And pathogens and insects can sense sometimes these volatiles and move or grow towards these compromised cells. Well, this fungus will actually grow on those compromised cells, this product. And we're talking about controlling botrytis, and our product is called Botrytis. So I think you know what it controls. And it's an antagonist that outcompetes nick occupation of ruptured cells. So as a preventative or as a quarantine preventative, you spray this out on the foliage and it will colonize and grow. However, too close to harvest, it may continue to grow. So we tell people, and again, we have programs for cannabis production on our website called the BioWorks Cannabis Program. And you'll read about when we tell you to stop applying these microbes based upon microbial testing that's being done. But this is a real good tool in the toolbox to control botrytis and other foliar issues by sheer competition for niche occupation.
So once I spot botrytis on a bud in a room, can I safely assume that the spore pressure in that room is already fairly high, or can I potentially catch it early enough and just remove? See, when you remove the bud, you're wounding the plant and I guess creating potentially other issues, another place for pathogen and disease. But what do you think? I mean, for people who can't apply microbials and can't harvest immediately, what other options really are there, if any, for preserving your crop?
It depends on. I've been in facilities, no names, but I've been in there where I saw a little bit of botrytis starting to creep up, and they cut it out and had no issues with that plant. Sometimes people will come back with some of these contact materials to help protect that wound, kind of like a temporary Band-Aid that can be done. Mm-hmm. I like to tell people, if you can, and it's small enough, try to cut it out. If it's getting out of hand, remove it. Then ask yourself, do I have a lot of air circulation here in this room? If I do, and the problem is botrytis, I may have spread spores. I better get out with some sort of contact preventative. And again, salt, soaps, and oils, I don't know, but I think they'd be tough to detect in testing. I just don't. I think they would be okay. Again, we're trying to, as a company, work closer with some of these testing labs to be able to give you a clearer answer on when these things can be applied. When's the last time they should be applied? So again, I know you're going to damage or maybe hurt a plant by trimming it, but if you can trim it a little bit, maybe come in with something to protect that, like an oil that wound, you could save that plant. But it's scouting. The key answer, Tad, to everything is scouting. Don't get lazy. Get out and look at your plants. And I know in cannabis, there's a lot of people out there and a lot of eyes, so your whole entire staff should be trained to go out and scout. And if they see something funny, report it. Just don't ignore it. So.
Yeah, and I want to highlight the importance of good genetics too. I mean, I've seen plants covered in botrytis and powdery mildew next to plants that don't even seem to care because they just have that innate resistance in them. And some people, they're like, this is just the best cultivar of cannabis I've ever grown. It's the one that everyone wants. At the end of the day, if it gets botrytis every single time, I probably wouldn't grow it. And a lot of people get stuck in that world of sort of that market need to grow something versus what's practical in their environment. So that's a challenge, but genetics is such a big role. At the end of the day, it really, I think, gets overlooked in a lot of ways. People are always trying to treat potentially plants that are genetically weak and trying to keep them healthy. And that's a real challenge.
Oh, I couldn't agree with you more. I think I said earlier, Tad, that, with the proper genetics, you really won't have too many issues. But if you do, your biologicals will work well because the pressure didn't get out of hand so rapidly. And I, like you, have been in facilities where there's different varieties side by side. One's getting ate up by something. The other one looks beautiful. I'll say, well, how come you're not growing more of this one? Well, this one what everybody wants. And I said, well, send it to your sister facility. You guys should concentrate on this variety because you know how to grow it well. I mean, yeah, genetics are so important, and you're going to see so much research be done on genetics in the next couple of years. It's going to be so nice.
So I wanted to jump back to powdery mildew. I had another question from a plant pathology perspective. We see, you mentioned fans and airflow, and a lot of growers are removing quite a few leaves, fan leaves, as a way of allowing better airflow through the plant, which in general I tend to agree with. Now, my question is sort of in terms of spreading powdery mildew. You mentioned that with Botrytis, those spores are riding in the air. What's going on with powdery mildew, and how much leaf tissue should we be removing in a given environment or a given canopy as a way of optimizing our plants' chance for success against these pathogens? Because when I see a space that's like when we get too many leaves or too dense of a canopy, and you dig into that canopy, not only can you not scout it, but you do see that's where the powdery mildew wants to live. The areas that aren't getting good airflow. So, what kind of recommendations would you have around sort of airflow and these pathogens?
Well, again, but powdery airflow's important. I mean, and so de-leafing can be a technique to help reduce the ability of powdery mildew to get a foothold. So I believe in de-leafing. Again, how much should you do? Well, do enough that you're getting good airflow, number one. Number two is de-leaf enough that you can scout in the middle of something, easier than having to crawl in there. So, I mean, de-leafing offers a couple of advantages. So, but powdery likes to spread more by water splashing on people's clothes. It's not blown around. It's not wind dispersed or air dispersed like Botrytis. So that's an advantage of keeping that airflow up. But again, with powdery, temperature is important. Get it up there. We talked about that at 75 or a little higher if you can. Powdery doesn't need free moisture. Botrytis does. Powdery you can get good airflow and stop it from getting a foothold. With Botrytis, it'll help disperse it. So there's no easy solution or answer, Tad. It's just, what's your biggest problem? Play around with airflow. Watch over fertilization. I mean, too much fertilizer can alter the metabolites being produced by these medicinal plants, but also it can weaken and stress a plant. We know that in ornamental plants, the flowering plants, if you add too much nitrogen, Pythium can attack the roots more effectively. Whiteflies can sense a plant is stressed and attack more effectively. In fact, and you guys have all heard about this, some of these so-called plant nutrition products offering the ability to reduce plant stress. When plants get stressed, they give off volatiles. And these volatiles are like gases. Ethylene is one that plants give off ethylene when they're stressed, and issues can sense and detect that ethylene emission, and especially insects, and go and attack that plant. So be cautious of too much cutting, too much damage to a plant. We have a plant nutrition called On Guard. And for lack of better terms, this amino acid based plant nutrition with a 500, for lack of better terms, I'm going to throw this out there, is I call it Prozac for plants. it reduces plant stress. And in addition, it is what I also call an Uber. It helps transport nutrients inside the plant more effectively and efficiently. So you can utilize less nutrients, save money, and not have salt built up. So there are ways to try to reduce stress in plants. And I know that University of Florida hired a biostimulant specialist a couple of years ago. His name is Dr. Shinsky Agahara, and he is working on the use of biostimulants because if you can reduce plant stress, plants are capable on their own of fighting off a lot of diseases and pathogens and insects, number one. Number two is they're not producing volatiles that help entice problems to come in faster. And if you can uptake nutrients more effectively, both your macro and your minor nutrients, the plant's going to be healthier, less stressed, and it can fight off a lot of problems on its own.
Yeah, I'm glad you brought up over-fertilization because I have seen what you're talking about there in relationship to nitrogen. Have you seen that sort of research done in an organic system where, say, that nitrogen may have been applied in the form of, fish meal or feather meal or some other form that's not immediately available or chelated, where you're waiting for the microbes to break it down into a plant available form? Are you seeing? I mean, I've seen where over application of those products will cause bacterial blooms, and also raise the temperature of the soil, which plants don't like, obviously. But are you seeing the same challenges around over fertilization in organic soils, I guess? Or have you seen research on that?
I've seen some research. Research is continuing, and there's a great deal of research out there on organic production, because one of the limiting factors in organic production is the availability of good organic sources of nitrogen that are either too available or not available, and your other needed elements. So work is being done. I can't sit here and quote you on any of it because a lot of it's still being conducted. Mm-hmm. I know here in Florida and the Southeast in general, I hang out a lot when I'm not in Mexico doing work, is there's more and more interest in some of these organic-based inputs. And in fact, we started a line a couple of years ago of biological fertilizers, we call them, where your nutrients are not readily released by moisture or temperature like a control-release fertilizers. You literally need a living soil with good biology to release the NPK, because they are released by microbes feeding on these dried granular materials and not by moisture and temperature like a lot of control releases are. So there's a lot of interest in this area. We as a company have interest in this area. We'd like to develop specific plant nutrients for the cannabis needs. And a lot of work is being done in this area. There are a lot of products out there, Tad. There are more products. I mean, darn, when I go to shows, I see a lot of stuff out there. And when I start asking specific questions, I don't get specific enough answers. I get a lot of features and benefits, but not a lot of science. So I will say there needs to be a whole lot more work done before we'll know what we need to be doing as far as proper plant nutrition, both in organics and in cannabis production.
I can't agree with you more. There are a lot of products out there, like you said, and new ones coming on the market every day. And it's really hard. One of the things that Suzanne and I have had long discussions on, and what she had me come down to the biocontrol conference to give a talk on, was specifically how do we evaluate or vet all of these products? You know, what are the questions as growers or end consumers do we need to be asking these companies to know not only that what we're putting on our plants is effective, but that it's even just as a low bar, just safe for the end consumer? Because there have been some companies in the past that have been less than honest with us about what they're putting in their product. And so there's some challenges around that too. But yeah, this fertilizer thing I think is really interesting. So I'm excited to see more research on the organic side come out in relationship to optimal nutrient levels associated with cannabis and what those effects are on some of these things like like you mentioned, like Botrytis and powdery mildew.
And I look forward to the day we can do more research to see how our products and other types of biologicals like ours affect metabolite production, because I think that is something that needs to be understood and measured.
Yeah, well, Chris, I know I know people are going to yell at me because you're a smart guy. I'm sure I didn't ask all the questions I should have asked. I'd love to get you back on here at some point to follow up, but I really appreciate your time today. And you explaining some of these different microbes and pathogens was really helpful. So thank you.
You're welcome, sir. And it's been an honor to talk with you. And hopefully I've helped a few cannabis growers out there understand a little bit better about the confusion that's going on. There's a whole lot of products out there being offered to you. Just ask questions, check them out. I mean, as a company, we have over 25 years of history in ornamentals and edible production, and I'd like to say we got all the answers in cannabis and hemp production, but we don't. We have questions. We want to work with those that would like to work with us to answer some of those questions, and that's the only way we're going to get things done. And as importantly, Tad, we need to make sure that we have some sort of system in place in the future to certify, or just to say these products have been shown to help, these haven't. Because like you said, there's a lot of products out there. There's a lot of fermented products. I don't want to see anybody get harmed by fecal coliforms or any anaerobic organisms, so be careful. Understand what you're doing. It may make sense to you. The person presenting the product may have made sense to you, but ask around. See what people are using. Check out the company's website. See what they can offer. If they can't answer a whole lot of questions, I'd have a whole lot of concerns about working with them.
I think that's great advice. Yeah. I appreciate that. And people, I will put links to your website, like I said, and some of the information that you mentioned on the podcast page so people can check it out there. And thank you very much.
Thank you, sir, for your time.
That was Chris Hayes with BioWorks, and you are listening to the Cannabis Cultivation and Science Podcast. I'm your host, Tad Hussey. You can check out the BioWorks website at www.bioworksinc.com. And don't forget to check out our website at www.kisorganics.com for more information and resources and links to the topics we discussed on the show today. Thanks for listening.